Hello, everybody. I'm Richard Tsien from NYU, and I'm going to talk to you today about orchestrating the brain for social behavior. We have some ambitious questions and goals facing us today. First, to delineate the goals of studies of the brain, computation, and behavior, and ask how they can be integrated with each other. To describe how oxytocin and other neuromodulators transform circuits in the brain and actually operate at a different level than fast neurotransmitters. And finally, to take a more meta view of the whole problem and evaluate the synergies and challenges of multi-level, multi-investigator projects, asking the question of how success in these projects can be defined. It just so happens that in this day and age of pandemic sheltering, 2 books crossed my desk in the last week. One was a copy of Principles of Neuroscience, the Kandel textbook, to which I contributed a chapter. And secondly, a book called Noise by Nobel Prize-winning Daniel Kahneman and his colleagues. These are 2 very different approaches to function of the brain. Principles is all about years of hard-won knowledge written about by many experts. Noise is a much more popular-oriented book trying to describe how human judgment is fallible and subject to large amounts of variation, both bias and variability. And the point is that if we could reduce that variability, we would be much better off whether we're trying to fight the pandemic or simply buy a used car. So my recollection of these problems goes back to the early days of being a graduate student and listening to a marvelous lecture by Colin Blakemore, which mentioned René Descartes' models of vision. René Descartes knew that an object in the real world got turned upside down in terms of its image on the retina. And he was worried about the fact that this image was upside down, so he arranged the optic nerve and its connections to what he thought was the seat of consciousness, the pineal gland, by having a re-inversion of that image so the image would project properly upright onto the pineal gland. Even as a graduate student, I realized that Descartes was simply kicking the problem down the road because how did the pineal gland know what to make of the image? And was the re-inversion by the nervous system an answer to the question, much more fundamental question of how we saw? Descartes had equally metaphorical ways of looking at memory. He saw memory as the imprint produced by a set of needles that could make little holes in a piece of canvas. And when the canvas was stretched out, the holes would become apparent and the memory could be recalled. So this is a metaphorical view both of vision and of memory. And as it turns out, these could be thought of as a hypothesis for how our brain processes information, stores it, and recalls it. Now, the topic for today is about oxytocin. This is a very interesting chemical, a 9-amino acid peptide produced in the hypothalamus of the brain and released centrally and in the periphery, for example, to trigger the milk letdown reflex. Oxytocin, consisting of 9 amino acids, is conveniently shaped like the letter 9, an easy way to remember that. And it's best known for its role in production of milk and childbirth. So it's really important for reproduction. but it's also implicated in many social behaviors. Social encounters between people and between humans and animals cause an increase in oxytocin that could be measured within a few minutes. So this is something that is highly relevant to social behavior. Oxytocin and its cousin, vasopressin signaling, was shown to be important for social behavior in rodents, in prairie voles and mountain voles, which differ radically in the amount of oxytocin receptor that they express, in classic experiments of Larry Young and Tom Insel. Oxytocin has also been tested as a possible clinical agent for treatment of autism, although the jury is still out on this matter. So oxytocin really exemplifies the logic of neuromodulation. The central nervous system uses fast transmitters, for example, glutamate, the excitatory transmitter, and acetylcholine, but it also has many neuromodulators, loosely defined as slower and less directly acting agents, which can be peptides or even lipids, and also small molecules like glutamate and GABA. Studying these molecules is extra challenging because we not only need to understand the neuromodulator, how it's produced and how it acts, but we also need to understand the basic circuit that it acts upon. Our hypothesis is that neural circuits function at multiple levels, and they can be transformed to have a different personality by the state of the neuromodulator. This idea was put forward for invertebrate circuits, for example, in the lobster by Yves Marder and others, but it's never been fully worked out in vertebrates. And this whole idea of modulating the existing operation of a circuit and turning it into a different kind of circuit is both powerful in concept and has interesting evolutionary aspects. Oxytocin is a particularly interesting exemplar, and our goal today is to derive principles generalizable not only to oxytocin, but generalizable to other neuromodulators and circuits. Now, in the brain, the receptors for oxytocin couple to downstream G proteins, and the receptors are found unevenly across the brain. One area where it's particularly prominent is in the hippocampus, in a particular subfield of the hippocampus called CA2. You can see that in this diagram, which shows CA2 principal neurons, pyramidal cells, lit up because they're expressing TD tomato under the control of an oxytocin receptor promoter. This indicates that CA2 is much richer in oxytocin receptors than its neighboring and more famous subregion called CA1. The oxytocin receptors in CA2 are particularly interesting because they have been implicated in social behavior in work from Steve Siegelbaum's lab, and also Raman Sahay showed that removing oxytocin receptors from these neurons caused a loss of social recognition. Memory driven by oxytocin was abolished. Furthermore, oxytocin receptors are also present in a particular small but very interesting region of the brain called the ventral medial portion of the hypothalamus and the ventrolateral part of that. So these two small regions are very important in Dayulin's work for controlling aggression, and they also happen to be replete with oxytocin receptors. So we obtained funding from NIH in the mechanism called the U-19, and my principal investigators, or co-investigators, are Rob Frumke, Yuri Buzhaki, Moses Chow, Annette Gray, Dayulin, Adam Marr, and Elisa Serkis. And we're all at NYU in the Neuroscience Institute and in the Skirball Institute. We are the only U-19 group of all the ones that have been funded that are working on modulation. And our institutional organization favors collaboration between us. Special thanks to our all-star cast of external advisors and to the NIH personnel listed on the first slide, who are both our advocates, critics, and teachers. Our U-19 grant, which is being presented here as an example of a successful large science collaboration, chose to focus on a number of regions of the brain that express the oxytocin receptor in abundance. Oxytocin is shown here, being released from the hypothalamus, and it acts on the auditory cortex, the VMHVL, the hippocampus, and the lateral septum. And as a result of the actions on the auditory cortex, you can see in the upper right-hand corner effects on pup retrieval. As a result of actions on the VMHVL, also in the hypothalamus, we see changes in aggression, but also changes in the recognition of one's social rank. So we have taken a multidisciplinary approach with this anatomical backdrop, looking at synaptic plasticity in the auditory cortex, looking at the transfer of information from hippocampus to the lateral septum, looking at the physiology of how CA2 principal cells respond to oxytocin and use various G proteins like GI and GO to control their ion channels and therefore their membrane potential and cellular excitability. And finally, looking at the ventromedial hypothalamus, where certain oxytocin receptor responding cells are really important for putting oneself in the totem pole of society. And of course, these four main projects are very well supported by cores that are involved in producing antibodies to the oxytocin receptor, in doing behavioral studies, in managing data and sharing data, and finally in administration and coordination, headed by Heather McKellar. So our principal investigators have many years of collective experience among them. And here you see some of the papers we published just over the last period of funding. Transformation of a spatial map across the hippocampal lateral septal circuit, transformation of the firing pattern of CA2 pyramidal cells, sexual dimorphic aggressive behaviors, and neuromodulation of maternal circuits by oxytocin. So I'm going to tell you about some very exciting work showing the involvement of oxytocin neurons in enabling the social transmission of maternal behavior. This is beautiful work coming from Juana Garcia, now a junior faculty member at Rutgers, working with Robert Frumke, one of our principal investigators. So now I'm going to show a video showing the behavior of an experienced mother who will come and retrieve her pup. So you will see her leave the nest, detect the pup who is giving out calls of distress, grab it by the scruff of the neck, and take it right back to the nest. Let's contrast that with the behavior of a female mouse that is not given birth and is inexperienced. And you will notice that the inexperienced mouse really pays the pup no mind, ignores its calls, and will just casually walk right past it without doing anything. Now, this is the behavior whereby the inexperienced animal becomes experienced with the tutelage of the mother, who has a vested interest in getting some babysitting help by teaching the naive female how to go back to the nest and how to gather pups. So in this behavior called shepherding, Frumke's lab noticed through 24/7 recordings that mothers would shepherd inexperienced females back to the nest time after time after time. And they recorded literally hundreds of these events. Now, notably, when the shepherding is occurring, the oxytocin neurons in the inexperienced female fire off madly and help it to acquire the skills that it needs. So this social transmission of maternal behavior consists, first of all, of oxytocin neurons firing in the virgin that is benefiting from the shepherding. That firing is critical for the acquisition of retrieval skills because if you block the oxytocin neurons from firing, the retrieval skills are not gained. The block of oxytocin receptors prevents the learning, and mimicry of the firing of oxytocin neurons actually helps promote the learning. So the pharmacological block of the oxytocin receptors by a very specific antagonist prevents the acquisition of behavior. So this shows in a very dramatic way the involvement of the oxytocin receptor system in social transmission of maternal behavior. So the practical strategies of our group are to combine top-down and bottom-up approaches. 24/7 recordings of animal behavior on one hand, analysis of oxytocin receptor distribution, signaling, and electrophysiological effects on the other hand. We do both of these approaches within individual labs. Our labs are pretty broadly spread within themselves, and we collaborate together. The behavior proceeds hand in hand with mechanistic analysis, and it also draws upon advances in labs outside of the U-19 project. For example, our colleague Yulong Li, working in Beijing, has done an excellent job of creating receptors for oxytocin, or optical receptors for oxytocin, that can monitor the local oxytocin concentration. We capitalize on our close physical proximity to each other, all within a large superblock of hospital. We synergize with other U-19 groups and other U mechanisms. For example, groups that are studying the role of oscillations in behavior in primates, other groups that are interested in synaptic plasticity and the role of brain oscillations. We combine experiments with computations and theory, and we try to push technological advances. For example, Yuri Buzhaki has been very active in studying ways of recording from hundreds of thousands of neurons, and Rob Frumke's lab is pioneering the use of caged oxytocin. And we use grant and paper writing as a way of training our diverse trainees. For example, my lab has many folks who are at the interface between our project, which is more about the biophysics and circuit mechanisms, and we collaborate together with Ishmael. Jingjing is involved in that with Rob Frumke's lab. We collaborate with the Buzhaki lab via his colleague. We collaborate with Dayu Lin's lab, studying both female aggression and social hierarchy. We collaborate with Paul Glimscher's lab, Kenwei Louis, and Bo Shen. And finally, we are actively involved in working with the Oxytocin Antibody Core and the behavioral optogenetics score. So all of these collaborations are driven by trainees. The trainees are very diverse, and we're very proud of their ability to progress. For example, Bianca Jones-Marlin got a job at Columbia. Katie Eyring won an F99 K00 and is now working with Dan Geschwin at UCLA. So to close, we have been successful in publishing a number of papers, including papers about plasticity in the auditory cortex, papers about the amygdala and its role in regulating aggressive and sexual behavior, papers about the routing of hippocampal oscillations to the lateral septum, and Yuri, the co-author of this, recently won the Gerard Prize of the Society of Neuroscience. And finally, my lab is interested in molecular and cellular mechanisms, and we study how the synaptic plasticity might be supported by synaptic vesicle traffic. So that ends part one, and if there were time, I would go on to part two, but I think I'm going to call it a day for today and simply end the presentation here, simply to add that Terry Sinowski's famous diagram about the many levels of brain investigation stand as a challenge to us, whether we are following the footsteps of René Descartes or not. And part of what we need to do is to take the abundance of genetic data on devastating disease and use it to help us understand how neurons work as individuals, how synapses communicate. Because many of the genes encode proteins involved in the action of individual neurons or indeed individual synapses. Only now are we beginning to comprehend how these genes and gene products are orchestrated to guide behavior, and we believe that circuits are a critical intermediate between cellular and molecular neuroscience, reaching down to the level of genes and proteins, and systems neuroscience ranging all the way up to the brain and behavior. So thanks very much for listening to me, and I look forward to any questions that you might send me. My email address is richard.tsien@nyumc.org. Thanks again.